Welcome, curious minds, to AI Paper Podcast.
Great to be here.
Today we're taking a deep dive into AlphaGenome, a really fascinating new paper that's truly pushing the boundaries of AI in genomics. Imagine a tool that acts like a crystal ball for your DNA, predicting exactly what's happening at the molecular level just by looking at a sequence. That's essentially what AlphaGenome aims to do. Okay, so for a quick rundown, with this groundbreaking AI, AlphaGenome can predict complex genomic features and genetic variant effects directly from these massive 1 megabase stretches of DNA. And it integrates all sorts of biological data, RNA-seq, ChIP-seq, ATAC-seq, even chromatin contact maps, and it works for both human and mouse genomes. What seems really special is its core architecture, a UNet-Style Design with transformers using something they call sequence parallelism for, really efficient computation. Plus, a key part is this two-pronged training. First, lots of pre-training on real experimental data, then this clever distillation where a student model learns from a whole ensemble of 64 teacher models. This lets AlphaGenome hit state-of-the-art accuracy, often significantly beating older models like Informer and Borzoi.
And it also gives us powerful tools like In-Silico Mutagenesis to really get why genetic change matters. So let's unpack how they pulled this off.
Indeed. What's truly impressive here is how they've managed to tackle the sheer complexity, the scale of the genome. They've managed to build a single, unified model that predicts this incredibly diverse range of genomic features. It gives you a truly comprehensive view, which is, it's no small feat.
Okay, so let's start with the grand vision then. What exactly is AlphaGenome? How big of a chunk of DNA are we actually talking about here? And what kind of biological signals can it predict?
Right. So AlphaGenome is designed to process an impressive 1 megabase. Yeah, that's 1 million base pairs of DNA sequence at a time. It also takes in whether it's human or mouse DNA, and its main goal to predict a massive number of genomic features or tracks. We're talking over 5,900 for human and more than 1,100 for mouse. These predictions cover lots of different cell types and 11 different categories of biological activity. Things ranging from measuring how active a gene is, like its expression, to identifying exactly where specific proteins bind to the DNA.
Wow, one member that's huge. For context, try and analyze that much data and predict thousands of things simultaneously. It feels like trying to listen to every conversation in a massive stadium at once and understand each one. How do they even begin to computationally manage that kind of scale? Does it just grind to a halt?
Yeah, that's a great question, and it's really where their architectural innovations shine. AlphaGenome uses UNet-Style Design. You might know this from image processing, but here it means it starts by condensing or downsampling the vast input DNA sequence. Then it processes it with powerful transformer layers. These can understand both local details and global relationships across the sequence. And finally, it expands those insights back, upsampling to a fine-grained resolution to make those precise predictions. But the crucial innovation for handling the scale is what they call sequence parallelism.
Sequence parallelism.
Okay. Yeah, they basically break the 1 millibase DNA sequence into smaller chunks, about 131 kilobases each. And then they process these concurrently across multiple computing devices like TPUs or GPUs.
Ah, okay. So it's kind of like a highly parallel assembly line for the DNA sequence, making sure it can handle the scale efficiently. And it integrates information both locally within a chunk and globally across them.
Precisely. That's a really good way to put it. This distributed computation is seamlessly integrated right within the UNet structure. So you have an encoder part that downsamples the sequence, captures these high-level embeddings. Then there's a transformer tower in the middle. This uses multi-head attention to establish global context, looking at pairwise relationships across the sequence at a coarser resolution, and then a decoder that upsamples these embeddings back to the original fine-grained resolution for making those task-specific predictions. It's a very elegant solution, really, for balancing local detail with global context over these really long sequences.
This sounds like some serious engineering. Okay, but having the architecture is one thing. How do you even train a model this complex? What kind of data do they feed into it, and what were the actual training methods?
Right, the training is also key. They use two main strategies to get this impressive performance. First, there's what they call pre-training. This is where the model learns directly from just a vast amount of experimental genomic data. It involved sampling continuous 1 millibase DNA intervals and then augmenting them.
Augmenting, what does that mean here?
Good question. It means they apply some transformations. like subtle random shifts to the sequence position, just shifting it slightly left or right, and also randomly applying reverse complementation, basically flipping the DNA strand and swapping the bases according to pairing rules.
Okay, so they're essentially teaching the model to recognize the underlying biological patterns, regardless of slight variations in position or which DNA strand it's looking at. That seems smart for making it robust in the real world.
Exactly. It makes the model more robust and effectively doubles the training data, too. And the scale of this pre-training was, well, immense. They used TPU v3 hardware, processing a batch size of 64 samples spread across 8 TPU cores using that sequence parallelism we just talked about. They trained for a fixed duration, 15,000 steps. And critically, they trained 64 different models this way, using all the available reference genome intervals. These became the teacher models for their second and maybe even more innovative strategy, distillation.
Okay, here's where it gets really interesting, I think. Distillation. For anyone listening who might not be familiar, what exactly is happening there? Is it kind of like a student learning from a panel of many wise teachers?
That's a wonderful analogy, actually. Yes. In distillation, a single student model learns to reproduce the predictions from those 64 frozen, meaning their parameters aren't changing anymore, pre-trained teacher models. But what's really clever is that during this distillation phase, they introduce additional input perturbations.
More perturbations, like what?
Things like randomly mutating a small percentage, maybe 4% of nucleotides, and also applying small structural variations, tiny insertions, deletions, and inversions with randomly chosen lengths.
Huh. So the student isn't just learning the original data patterns, but it's also learning how to handle subtle changes, like noise or small mutations, by mimicking how the ensemble of teachers robustly responds to those variations. That's clever. And what's the big advantage of doing all this, practically speaking?
Well, the practical implication is profound. Distillation allows them to achieve performance that's competitive with, or sometimes even better than, using the large ensemble of all 64 teacher models directly. But, and this is the key part, it does so with significantly reduced inference costs.
So it's much cheaper and faster to actually use the final model.
Exactly. A single student model effectively captures the combined knowledge of many teachers. This makes it much more efficient for deployment. You know, we're talking about making complex, state-of-the-art genomic analysis potentially feasible, not just in huge research labs, but maybe closer to clinical settings. It could make personalized medicine much more practical by cutting down computation time and resources drastically.
Wow, okay. That really highlights the potential impact.
Yeah. And the distillation training itself was also a massive undertaking, just to give you a sense. It ran for 250,000 steps on 64 NVIDIA H100 GPUs, with each GPU loading a different frozen teacher model to learn from.
Okay, so a huge effort on the training side. Now, you mentioned earlier that AlphaGenome predicts 11 different output types across thousands of these tracks. What kind of raw biological data are they actually predicting and learning from? Processing and integrating all that sounds like a massive job in itself.
It truly is a data integration marvel. They had to assemble just a vast array of publicly accessible data sets to feed this model. This includes things like RNA-seq, which measures gene activity or expression across different tissues and cell types, data from ENCODE and GTEx, for example, also CAGE and ProCap data, which help pinpoint exactly where genes start being transcribed. And then for chromatin features, how the DNA is packaged, they use DNA-seq and ATAC-seq. These tell us which parts of the DNA are physically accessible to the cellular machinery. Plus, ChIP-seq data, which identifies where specific proteins like transcription factors or histone modifications bind to the DNA. And they even include chromatin contact maps like Hi-C, which show how distant parts of the genome physically loop around and interact with each other in 3D space.
So it's really not just looking at one piece of the puzzle, like gene expression alone. It's trying to see the whole biological system at once, integrating all these different layers of genomic information.
Precisely. That's the core idea. And for each data type, they had to apply really rigorous quality control and normalization steps. It wasn't just plug and play. For example, RNA-seq data is normalized to reads per million, or RPM, to allow fair comparisons between different experiments. They applied strict quality filters. And for the accessibility data, like ATAC-seq and DNA-seq, They actually ingested the raw alignment files, the BAM files, to convert them into base resolution count files themselves. This preserves very fine-grained information and let them apply specific corrections, like accounting for how the enzymes they use actually cut the DNA. The key takeaway is really the meticulous data preparation required to integrate such diverse data cleanly.
Okay, that makes sense. Now, one thing that stood out to me reading the paper was how they handle something as incredibly complex as gene splicing. It feels like a whole different ballgame from just measuring overall expression levels. How does AlphaGenome tackle that?
That's an excellent point. And it is complex. For the splicing-related predictions, the data actually comes from the same RNA-seq datasets we mentioned, but they apply a very, very stringent processing pipeline using tools like STAR for alignment, and then a quality filtering approach from a package called SpliceMap. This compiles lists of high-confidence splice junctions, basically, the points where the RNA molecule is cut and then stitched back together during splicing. These retained junction counts are then normalized and pre-processed, often represented as like a matrix showing connections between potential donor and acceptor splice sites. This structured data allows the model to learn the incredibly intricate rules and relationships that govern how genes are spliced, which, as you know, is crucial because a single gene can often produce many different protein variants through alternative splicing.
Right, okay. So they put a lot of effort into preparing that splicing data specifically. Now, with all this amazing architecture, the complex training, the meticulous data integration, how well does it actually perform? Does AlphaGenome live up to its ambitious name when predicting real biological phenomena?
The results reported are very strong, yes. AlphaGenome shows significant relative improvement over the best competing models across a wide range of tasks. That includes both the genomic track prediction tasks we discussed, like predicting expression or accessibility, and also for predicting the effects of genetic variants. This relative improvement metric they use is quite useful because it contextualizes the gains. It normalizes AlphaGenome's performance against how much a comparison model, like say Informer, improved over just a random baseline prediction.
So it tells you not just if it's better, but how much better it is relative to existing progress.
Exactly. It really highlights the impact of the improvements, not just the raw numbers.
Okay, so how much better are we talking? Specifically for people working in this field, what do those percentages actually mean in practical terms?
Well, for the genome track predictions, AlphaGenome showed relative improvements anywhere from, say, 1% up to over 30%, specifically 32.6% in some cases against leading models like Borzoi or Informer. This varied depending on the specific data type and the resolution, but it means for many tasks it's not just a tiny bit better, but substantially more accurate. While some raw comparisons, maybe for RNA-seq at very high resolution, might look different due to how things are measured, when you look at gene-level activities or transform the data appropriately using logs, AlphaGenome consistently shows much better agreement with the experimental results. And importantly, it also significantly outperforms models that were specialized, built only for specific tasks, like ProCapNet just for ProCap data, or ChromeDPNet just for ATAC and DNase data across multiple metrics.
That's impressive, beating specialized models with a general one. And what about something really challenging, like predicting the impact of just a single genetic change, a single letter difference in the DNA? That's so crucial for understanding disease mechanisms and for personalized medicine.
Yes, and this is an area where AlphaGenome really seems to excel based on their benchmarks. It demonstrated superior performance on various variant effect prediction tasks. This includes predicting the impact of variations on chromatin accessibility, things called DSQTLs or KQTLs, also identifying unusual splicing events, sometimes called splicing outliers, which can be linked to disease, and accurately estimating the effect size of known EQTLs. Those are genetic variants already known to influence gene expression levels. Its splicing predictions, specifically when compared against other specialized models like Splice AI or SPLAM, showed really strong performance, like high OPRC scores.
OPRC area under the precision recall curve.
Exactly. High OPRC indicates it's good at correctly classifying true versus false splice junctions and predicting how much different splice sites are actually used. This really feels like a massive leap forward for understanding how genetic variation contributes to disease at the molecular level.
Okay, so it's good at predicting. But predicting is one thing, right? Understanding why a variant has a certain effect or figuring out what part of the DNA sequence is actually important for that prediction, that feels like a whole other level of understanding. How does AlphaGenome help researchers get to that deeper insight?
You're absolutely right. Interpretation is key. And this is where AlphaGenome's Genome Interpretation Suite comes into play. It's a set of tools built around the model. A core part of this is distilling the model's multifaceted genomic predictions, all those thousands of tracks, down into a single informative variant score for each genetic variant you're interested in. This score basically quantifies the predicted biological impact. It measures the difference in the model's predicted features between a DNA sequence carrying the original or reference allele versus one carrying the alternative or mutated allele.
So it's essentially trying to tell you, okay, this specific change, this A to a G at this position, is likely to do this much difference to that set of biological processes, and it gives you a single number to quantify that. That allows for comparison.
Exactly, a single score summarizing the overall predicted functional difference. But raw scores can be hard to compare across different types of predictions or different regions of the genome. So to make them more interpretable and comparable, they apply what they call an empirical quantile calibration.
Quantile calibration, how does that work?
Well, it involves comparing each variant score against a large background set of common human genetic variations. They used over 340,000 common SNPs from chromosome 22 as a reference set. By seeing where a specific variant score falls within the distribution of scores from these common variants, they derive a quantile score. This standardizes the predicted impact. It tells you if this variant's predicted effect is, say, in the top 1% compared to typical variation, or maybe just average. It makes it much easier to flag potentially high-impact changes.
That sounds incredibly useful for researchers trying to prioritize variants. Okay, and then there's this technique called In-Silico Mutagenesis, or ISM. That sounds like a really powerful tool for dissecting exactly what's going on in the sequence. How does ISM work with AlphaGenome?
ISM is indeed a very powerful technique for interpreting which specific nucleotides contribute most to AlphaGenome's predictions for a given region. It works by systematically predicting the effects of all possible single nucleotide changes within that sequence region. So for each position, it checks what happens if you change it to an A, a C, a G, or a T. By comparing the model's output for the original sequence versus these mutated sequences, it estimates a contribution score for each base position. This reveals which bases are most critical for a given predicted outcome, like gene expression or protein binding. These scores are often visualized as sequence logos, you might have seen them, where the height of each letter, A, C, G, T, at each position reflects its calculated contribution. It's almost like a heat map showing you the hot spots of functional importance in that piece of DNA.
Right. So using this ISM, you could literally see if a known disease-causing variant falls right on a base that ISM highlights as being super important for, say, a transcription factor binding.
Exactly. Or even more powerfully, you can use comparative ISM.
Comparative ISM.
Yeah. By applying the whole ISM procedure independently to both the original reference sequence and the sequence containing the alternative allele, the variant you're interested in, you can directly compare the resulting contribution score profiles side by side.
Ah, I see. So you can look for differences in the importance maps.
Precisely. This powerful comparison helps you identify if the variant, for example, disrupts an existing important regulatory motif that was present in the reference sequence. Or, conversely, maybe it creates a novel motif, a new important site, in the sequence with the alternative allele. This is absolutely critical for getting at the precise molecular mechanisms underlying genetic variations and understanding their role in biology and disease. It moves beyond just saying this variant has an effect to potentially showing how it has that effect.
That's fantastic. It really bridges the gap between prediction and mechanistic understanding. Okay, so every great model, every big project like this has its secret sauce, right? Mm-hmm. After doing all this development and testing, what did the researchers learn about what truly makes AlphaGenome so powerful and perform so well? What were the key takeaways from their own experiments, like the ablation studies?
Yeah, they did conduct extensive ablation studies. That's where you systematically remove components or change training strategies to see what impact it has on performance. It helps you understand what's really driving the success. And two key takeaways truly stood out from their results: the significant benefits of distillation, which we talked about, and also multimodal learning.
Okay, we covered distillation earlier, mainly focusing on how it makes the model much more efficient for deployment, for inference. Did the ablation studies show that it also improved the model's raw performance itself?
Yes, interestingly it did. The study showed that the single student models produced by distillation, especially when they learned from a large number of teacher models, like the 64 they used, actually performed competitively with, or sometimes even surpassed, the average performance you'd get from using an ensemble of several independently pre-trained models.
Wow. So you get the efficiency and potentially even better performance.
In many cases, yes. It means you can achieve really strong performance with significantly reduced inference costs compared to the standard practice of evaluating large ensembles. Even just distilling from a single teacher model showed benefits for some tasks. So distillation wasn't just about efficiency, it contributed to the robustness and accuracy too.
Okay, that's a really important finding. And the second key takeaway was multimodal learning. What exactly does that refer to in this context, and why do they find it was so important?
Right. Multimodal learning here refers specifically to the integration of all those diverse biological data types during the training process, the RNA-seq, the ChIP-DSEq, ATAC-seq, the contact maps, all it together. And their ablation studies clearly showed that the fully multimodal model, the one trained on everything, generally outperformed models that were trained only on a single type of data, or maybe just a subgroup like only accessibility data. This really confirms the overall benefit of integrating diverse data types. It seems to allow the model to learn shared, richer, more comprehensive representations of genomic function.
So it's like connecting all the dots from these different biological layers actually helps the model understand each individual layer better, even if the primary prediction task isn't specifically about combining them. That seems to echo how biology actually works, doesn't it?
Exactly. That's a great way to think about it. The benefit was sometimes task-dependent, of course. For example, they found that predicting accessibility variance could be done quite effectively using only the accessibility data group. But for more complex tasks, like predicting the impact of variance on gene expression, EQTLs, the full multimodal input, incorporating all the data types, was absolutely crucial for top performance. It really emphasizes that biology is this deeply interconnected system, and the model seems to benefit significantly from reflecting that underlying complexity in its training data and architecture.
That makes a lot of sense. So wrapping things up, what does this all mean for you listening, whether you're an AI researcher, an ML engineer, or maybe someone working more directly in the biological sciences? What's the big picture here? AlphaGenome feels like more than just another predictive model. It seems like a powerful new lens, doesn't it? A new way to understand the incredibly complex language of our genome. From predicting these intricate biological signals across vast stretches of DNA to pinpointing the likely impact of very subtle genetic variations, is providing, well, potentially unprecedented insights and efficiency. And it feels like it's moving us closer to that goal of truly personalized medicine.
Absolutely. And this capability, this power, raises an important question, maybe something for you, the listener, to consider. As models like AlphaGenome become increasingly adept at interpreting the genome at such incredible scale and detail, what do you think are the most impactful scientific or perhaps clinical applications that will emerge from this? And maybe even more excitingly, what entirely new research questions will we finally be able to ask? Questions that were simply out of reach before we had tools like this.
That's definitely food for thought. What new doors does this open? Well, thanks for joining us on this deep dive into AlphaGenome here on AI Paper Podcasts. We really hope you've gained some valuable insights, and maybe you're as excited as we are about the future unfolding at the intersection of AI and genomics. Thanks for listening.